Welcome to lecture 13 in DIY transcriptomics. At this point in the course, you've already developed a really strong foundation in bioinformatics by learning to analyze bulk RNA-seq data. So in this lecture and the next lecture, we're going to build on that foundation with new skills for analyzing single-cell RNA-seq data. Now, as we make this transition from bulk to single-cell, I want you to keep in mind that a lot of the tools and concepts that you have already become familiar with will absolutely feature prominently in these lectures and in dealing with single-cell RNA-seq data. We're still going to have to carry out read mapping. There'll be some filtering. We definitely have to think about normalization. Dimensional reduction will feature very prominently, and we'll need to identify differentially expressed genes. So at a high level, we still need to go through these steps. But because single-cell RNA-seq data, and in fact any new data type, has its own nuances or features that make it unique, we have to think a little bit about exactly how we do these steps. I also want to just remind you that you have this really great toolkit with R and Bioconductor that is absolutely going to be essential for you in being successful analyzing single-cell RNA-seq data. Okay, so if we click on the lecture card for today, I want to take a minute just to review our learning objectives. I want you to understand droplet-based technology and how it allows us really to carry out not just single-cell RNA sequencing, but in fact, many different kinds of assays on single cells. You'll be able to compare and contrast single-cell and bulk RNA-seq methods by the end of these lectures. You'll understand a bit about the cost and experimental design considerations for planning your own single-cell RNA-seq experiments. I want you to get some familiarity with what we'll call multiplexed single-cell assays, things like site-seq and T-seq. You'll be able to define common terms and concepts in single-cell genomics, as you can already do with bulk cell. And then finally, we'll conclude this lecture by actually using a tool related to the Callisto aligners and developed by the same group called Callisto Bus Tools to carry out read mapping and pre-processing of raw single-cell RNA-seq data directly on our laptops. Okay, so before we jump into the details of single-cell RNA-seq data, I just want to take a minute to hopefully impress upon you that the field of single-cell genomics is developing exceptionally quickly. It's just exploding. And I think that's really nicely illustrated in this plot where we're looking at time since 2002 to current and the total number of cells profiled in a single cell experiment. And each dot here is a single cell experiment from the literature. And this data, by the way, the source, the publication associated with this data and the actual database where this data was pulled from is referenced here at the bottom of the slide. But one thing I hope you can see is just the frequency, the sheer number of single-cell RNA-seq experiments in the literature has just exploded. Lots more dots here. And we see this just incredible rise in the number of cells that people are profiling in their papers. The other thing I want to point out here, and it's illustrated by the coloring, each point here is colored based on the technology used to generate the single-cell data. And you see predominantly two colors dominating this graph, indicating the SmartSeq technology and 10x genomics technology. And what you can see is in the early, and I use this term relative as a very relative term, early years of single-cell sequencing being 2015, most of the experiments were carried out using SmartSeq technology. But more recently, in the last 5 to 7 years, there's really been an explosion in the use of 10x genomics technology for profiling single cells. So for the purposes of this lecture, we're going to focus on 10x genomics and their Chromium controller. That's not to say that SmartSeq isn't really incredible technology, and it's not to say that other newer technologies won't feature really prominently in the single cell landscape. This just means it's a good place for you to start when you're learning about single-cell data because it's a very popular platform and one that's responsible for generating a lot of the data you're going to see in publications. So one thing that's not really illustrated on this graph is the kinds of tissues people are profiling. And I think this is interesting to look at. If we replot this data based on the total number of experiments by tissues, what you can see, and perhaps not surprising, is that the most frequently profiled tissue is the brain. Why? Probably because the brain, we know it's extremely complex and made up of many different cell types, but we also know that some of the cell types that are the most important are also the most infrequent. And so it becomes a signal to noise issue with bulk RNA-seq data. If we grind up and extract RNA from a whole brain, we might lose the ability to detect those rare but important cell types. And so for the purposes of brain research, single-cell RNA-seq becomes really powerful. You can see it's been applied to a lot of culture methods, blood, lung. I hope the other thing you take away from this is that there's still a lot of tissues and therefore a lot of biology that remains to be explored because they really aren't well represented in the landscape of single-cell experiments to date. All right, I told you we would focus on 10x, so I want to take a minute to just explain that technology. 10x Genomics has made this instrument called the Chromium controller, very small, smaller than a desktop printer, and it has a single drawer that ejects, and in that tray, if you will, you set a chip. And that chip, although it resembles a sort of small, like 96 well plate in the sense that it has these rows and columns of wells, it's more than just a 96 well plate. It's really a microfluidic chip because underlying each of those wells is a microfluidic device. And so let's talk about how this works and how you go from this chip to droplets containing single cells. In the chip, you load your sample, so your cell suspension, along with master mix. And that master mix contains a lot of the components you're probably already familiar with from molecular biology. It contains reverse transcriptase, dNTPs, lots of components that are familiar to us. In the second row, you load gel beads, and we'll talk more about these beads in just a minute. And then last but certainly not least, we add oil to the top row here, and that oil is essential to actually allowing us to form droplets. Now, each sample corresponds to a single column here, so let's focus in on that column. As you load this chip into the Chromium controller and it takes the tray in, it'll apply a pressure plate to the top, forcing these components to the bottom of the wells and into the microfluidic device that underlies those wells. And as that happens, the beads, indicated here, begin to flow together with your cells and the aqueous solution or aqueous master mix that was in this first row. And as that combination of cells and bead and master mix occurs, they pass through a perpendicular flow of oil that you had loaded here. And that oil stream effectively pinches off tiny droplets and forms these droplets that you end up capturing from the Chromium controller. Now, the physics and fluidics of the flow rate of your beads and your cells and the oil determine the frequency of these droplets. And of course, the concentration of your beads and your cells and other reagents determine how frequently your droplets contain a bead and a cell, or a bead alone, a cell alone, and hopefully not, but multiple cells inside of a droplet, which is something we want to avoid. One advantage of the 10x platform is you don't have to fine-tune any of these parameters of the microfluidic device or the flow rates. This all is taken care of for you on the Chromium controller. So it really is pretty push button. If we take a closer look at one of these droplets, we can see how these components come together. Here's our bead, our cell, the master mix aqueous solution, and then outside of this droplet is the oil, which prevents this droplet from merging or mixing with an adjacent droplet. So if we just take a look at a cartoon schematic of what I just described, and this is by no means meant to represent what a microfluidic device looks like on any platform, but the basic idea is the same. We have our cells, our master mix reagent and beads all flowing together, and that perpendicular flow of oil colliding with that stream of reagents to pinch off droplets. And really what's happening here is each of these droplets now effectively becomes a reaction vessel, much in the same way that an Eppendorf tube or the well of a 96 well plate is a reaction vessel. This is a reaction vessel just on a much smaller volume scale. So this is actually what that process looks like. And again, the technology platform is not particularly important, just the concept that we have a flow of aqueous reagents, and that actually includes cells, a perpendicular flow of oil that's colliding with that the aqueous flow and resulting in a pinching off of droplets that then flow to the right. Similarly, we can see beads inside the video and the droplets in the video below. Again, droplets preventing from merging with each other because they're in this oil emulsion, right? So the contents of this bead remain separate from the contents of that bead. Once a cell has been captured in a droplet with a bead, the reagents inside that master mix cause the cell to lyse and cause this bead to dissolve, because remember, the bead is made of a gel substance. And so I want to take a minute just to highlight what's going to happen on the molecular biology end here, because those beads were conjugated to these oligonucleotides, and they have a few features that are really key to pointing out. The first is they have a constant, what we'll call read one sequence of 22 nucleotides that doesn't change across different beads, different droplets. But they do have a barcode here of 16 nucleotides that is unique to this bead. And assuming we've captured a single cell in that droplet with a single bead, then this barcode tells us about all the transcripts that came from that lyse cell in that single droplet. Okay, so this is really unique to each bead, and hopefully that means each cell if we captured only a single cell in the droplet. We then have a 10 nucleotide unique molecular identifier, or UMI, and that UMI is unique to each molecule. So that'll tell us specifically about this molecule that was captured from the cell. Another mRNA floating around in this droplet would have a different UMI. Okay, so once that process has happened, the rest of the things that occur are things that are probably already pretty familiar to you. Again, we have our gel bead. Here's the capture oligo. We have a poly-T sequence at this end, which of course serves to capture that poly-A, 3' poly-A tail in the mRNA. We can then begin a reverse transcription reaction because there's RT enzyme in that master mix that flowed into that droplet. It carries out the RT reaction, and as is so often the case, when these RT enzymes get to the end of the mRNA template, they will add untemplated Cs at the 5' end of the transcript. When that happens, that serves as a substrate for binding of what's called the template switch oligo, or TSO. And as that TSO binds, the RT enzyme will then extend a copy of that switch oligo. And this gets us to a point we always want to get to at some point in library preparation, which is a situation where we have a known sequence or handle at one end and a known sequence at the other end. Once we have that, then we can begin to do things like PCR amplification. So just to highlight at this point, we have created a cDNA of our originally captured mRNA, and that cDNA has a barcode that tells us which cell this transcript came from, and a UMI that uniquely identifies that molecule. At this point, because of these identifiers, the droplets could now be broken and the remaining steps can be carried out in bulk. Because we now have these identifiers in place, we no longer need the droplet. It's performed its function. As I said, those known sequence handles at each end of the cDNA now serve as the basis for PCR primers to amplify our cDNA. And as we often do in library preparation, we can do a quick QC on a tape station or other instrument for sizing molecules. And we can see, hopefully, if our first steps of the library prep worked, we get this nice distribution of cDNA sizes corresponding to different sized mRNAs that were captured. We then carry out an enzymatic fragmentation that's going to cut at random in this area. And that's important because it means we generate 1, shorter fragments, and 2 fragments that are 3' biased, because we're losing some portion of this 5' end. And that's why this library prep, which is usually the most sort of common single-cell RNA sequencing library prep, is considered a 3' biased single-cell RNA-seq prep. We then carry out end repair, A-tailing, and ligation to get us back to known sequences on each end. Clean that up, and this time we add primers that have both the Illumina P7 and P5 sequences. That's what allows the product of this PCR to bind to the Illumina flow cell for sequencing. But we also include a sample index that uniquely tags each sample that we're working with. We can then, after final PCR, check our libraries again, and we should see a tighter distribution and one that's smaller in size. Instead of around 1,000 or 1,500, we're around 400 or 500, and that's because, again, we fragmented here. Okay, so once we have our library and we begin sequencing, it's a little bit of a different situation from what we're familiar with from bulk RNA-seq. With bulk RNA-seq, we would be totally fine with single-end 75 base pair sequencing. That gives us enough information about the transcript to carry out read mapping with pretty high confidence. But with single-cell RNA-seq, we have to do paired-end sequencing. And the reason we have to do that is we need one read just to capture this critical information about the barcode and the UMI that tell us where what cell that came from and what molecule it is. It doesn't need to be a long read. About 30 nucleotides is sufficient, but we need that information. We then carry out a reverse read that actually comes off of this TrueSeq read 2 sequence and captures the information about the transcript. Without this barcode and UMI, this read is not useful, so they really need to be paired-end sequencing. One thing that's critical to understand is that this idea of using these oligonucleotides that contain different kinds of barcodes or identifiers and poly-T capture sequences really represents a very flexible way to build new single-cell assays. And I think one great example of that, and one I just want to highlight, is the development of something called CiteSeq, and that stands for Cellular Indexing of Transcriptomes and Epitomes by Sequencing. And the way CiteSeq works is instead of a bead conjugated to an oligo, it uses an antibody conjugated to an oligo. And that oligo contains a known PCR handle sequence, a barcode that is unique to that clone of antibody, and then a poly-A sequence. Now, this antibody barcode, I said it's unique to the clone of antibody. So if this were an antibody against, say, CD4 on the surface of CD4 T cells, it would have its own specific barcode. But if we had another antibody that was specific to macrophages, a molecule on the surface of macrophages, it would have a separate barcode. Now, this poly-A tail means that this whole oligo looks, at least to a capture bead, like an mRNA. And so the way CiteSeq works then really isn't that all that different from what I already described to you. So it shows you how you can develop new assays on these droplet-based platforms pretty easily. So in the case of CiteSeq, you take your intact live cells, you bind or incubate them with antibody that will recognize the markers, and you could do this with multiple antibodies because we have these unique barcodes for each clone of antibody. You then flow those cells into the droplet-based platform that we already talked about. And if you capture a cell that's labeled with one of these antibodies, as the cell lyses, it releases its mRNAs, and it also releases this surface oligo that looks like an mRNA. That bead with the poly-T sequences on it will capture the mRNAs and the antibody tag, but we'll always be able to tell apart that antibody oligo because it has that antibody barcode. And so that allows you to carry out in the same cells single-cell mRNA-seq and single-cell proteomics for surface proteins. And so in the case of the original CiteSeq paper, you can see that if you look at CD3 and CD4, CD8, CD56, you see really good agreement between the mRNA-seq carried out at a single-cell level and the antibody-based detection. But in some cases, like we see with CD8, we often get better signal with protein on the surface than we did with transcript in the same cells. And so CiteSeq is a great example of how we can multiplex single-cell assays and how they can be both complementary and also change the way we think about identifying clusters of cells. We could use the CD8 protein to unambiguously identify CD8 cells and then look at the transcriptional profile of those CD8 cells. This idea of using bar-coded antibodies has been commercialized, and maybe one of the most popular resources for that is the TotalSeq line of antibodies from BioLegend, where they have a whole range of mouse and human antibodies that can be used together as a cocktail, because again, they're all uniquely bar-coded, to really give you really rich proteomics data in conjunction with your single-cell RNA-seq data. This idea of multiplexing assays or using the droplet technology in really versatile ways extends far beyond site-seq. In fact, you can, instead of flowing whole cells into the fluidic devices, you can use just nuclei. And that's important because in some cases, tissue, even if it's fresh, it can be hard to get some cell types out of the tissue intact. And certainly if the tissue is frozen or if it's formalin fixed and paraffin embedded, it can be impossible to get intact cells out. But you can always disaggregate the tissue and get out nuclei, and those nuclei can be encapsulated just as we already saw for intact cells. Now, because these are nuclei, we can actually include in our master mix transposase that can cut open regions of chromatin and carry out a TAC-seq at single cell resolution. I already mentioned that single-cell assays can be multiplexed, and I think there's maybe no better example of that than a paper earlier this year that described something they called T-seq, which allowed profiling simultaneously of transcripts, epitopes, and chromatin accessibility, so TEA, in single cells. And the way this works is probably pretty familiar to you by this point, based on what I've already told you. That you can take whole cells, you tag them with antibodies with the oligos conjugated to the antibodies, so this is site-seq. You then permeabilize the cells and flow in a TN5 that will make it to the nucleus and cut accessible areas of chromatin. And this all happens in droplets. Then when the cell is lysed, you get capture of your mRNAs, capture of your antibody tags, and capture of the accessible chromatin regions as well. Now, just as a comment here, these kinds of methods obviously can take more optimization at the bench, but many of them are being commercialized and are available as kits. However, the data that results from these, keep in mind, you're getting transcriptional data, you're getting protein data, and you're getting chromatin accessibility data. The methods for integrating and analyzing these data are probably one of the most complex in terms of the whole single cell workflow. All right, now everything I've told you really is about single cells in droplets. And when we disaggregate tissue and flow single cells into droplets, we lose all information about where those cells were in the tissue. And so one of the really exciting areas of single cell RNA sequencing has been this push to develop methods to preserve spatial information about where those transcripts came from, which cells they came from, but also where those cells were located in a tissue. Of course, if we take a spleen and we mash up that spleen and carry out single-cell seq or site-seq or T-seq, we can get a lot of information about the T cells and B cells in that spleen, but we could never get information about where they are. And that location information is really critical to the biology of these different cell types. We know in an actual spleen you have germinal centers where these cell types interact that determines the outcome of an immune response. And so one of the big technology developments, and something that's changing really rapidly and that will continue to change over the coming years, is an array of technologies based around trying to localize transcripts in these intact tissues. And so conceptually, that's not all that different than what we talked about, the technology hurdle that Illumina struggled with, where they were trying to figure out how to amplify molecules on a solid surface, ultimately doing that through bridge amplification. The same concept applies here to tissue. We'd like to take transcripts that are too hard to image or visualize or sequence in a tissue and build them up so that we have enough signal to detect them in the tissue. And so methods have already been developed that really get to this problem. And one, also based on the 10x Chromium controller platform, takes advantage of the fact that we can just change the sequence on these beads to include a spatial barcode or a zip code. The beads, instead of then being free-flowing and flowing into droplets can be arranged in a pattern on a glass slide that you can then lay a tissue section down on. When that tissue section gets permeabilized and the mRNAs are available for capture, then you have captured not just the mRNA, but you've identified exactly where on this slide it came from. And so that kind of spatial information lets you then localize, in this case in the brain, expression of specific genes to, for example, the hippocampus. Now, resolution here is an issue. How big this spot is and how many cells could potentially give rise to, for example, this HPCA expression is just one of the technology limitations at the time, at this current time. But there are other methods out there that can allow or enable true single cell resolution. One such method called fluorescent in situ sequencing, or FISSEQ, really probably will be recognizable to you, at least in concept, from what we discussed in the pre-course lecture, where the goal here, as I said already, is to immobilize a transcript in its location in the tissue and then build up the signal from that transcript. And so you can do that in FISSEQ by creating an amine-modified cDNA, sort of cross-linking that to the tissue, circularizing it, and then using a high-fidelity, highly processive polymerase to make many, many copies of that circularized transcript in that one region. You can then cross-link those in that region so they stay there. And if you have incorporated fluorophores into this, then you could image and hopefully detect the original mRNA in its amplified form and know what cell type it came from. And this has been quite successful. There are drawbacks or challenges with this technology, like there are with any technology. And in the original FISSEQ paper, the authors highlighted that most users will need to reserve the confocal microscope for 2 to 3 weeks so that sequencing can proceed uninterrupted. Obviously, many of us would get into a lot of trouble if we took up a microscope in our core for 2 to 3 weeks. But as you can imagine, this technology now is being optimized and really divorced from a traditional confocal microscope so that it's a standalone piece of hardware. And so I expect we'll see lots of exciting developments in spatial single-cell transcriptomics in the years to come. All right, so before we conclude, I just want to highlight that now we are in a position in the field where transcriptomics includes not just bulk RNA-seq, but single cell analysis and now spatial transcriptomics. And these single cell assays can be easily multiplexed to give us rich information about each cell. It's important to know that one is not better than the other here. All of these approaches have their own drawbacks and own strengths, and they actually complement each other in ways that I think are important for you to understand. So, for example, in a single cell experiment, when we identify transcripts or genes unique to a specific cell type, we can use that information to inform our bulk RNA-seq analysis. And this is actually a really powerful thing that you can do. You can take single cell data to take a bulk RNA-seq data set and deconvolve it to try to understand the kinds of cells that might be generating your bulk RNA-seq data. Is it primarily CD4s or CD8s? What the relative contribution of different cell types might be to that bulk data. These bulk RNA-seq data sets, particularly those carried out on highly enriched sorted cells, then provide really important references for understanding clusters of unknown origin in single cell data. So if I have a cluster of cells and I don't know what those cells are based on 1 or 2 markers I might have in my mind at the time, I can use a large collection of bulk RNA-seq data from pure cell types and ask if any of those bulk RNA-seq signatures match well with this cluster and therefore give me a sense of that cluster's identity. And then finally, single cell data really helps us fill in some of the sparsity of spatial transcriptomics data to help us identify additional cell types in tissues. Okay, with that, we'll end and we'll pick up in just a minute.